Welcome to Chapter 3F. In this segment, we'll explore the kinetics view, an important tool in HDX analysis that examines the rate of hydrogen-deuterium exchange in proteins. By tracking these kinetic changes over time, you gain valuable insights into protein dynamics, interactions, and function within biological systems. Let's dive in.
Another view available here on the right-hand side is the kinetics plot. As with any other panel here, it is something that you can completely customize. You can drag it to any part of your screen. You can put it on a second monitor, or you can dock it somewhere else in the UI. So the kinetics plot contains a few options. And if you don't see all of the options in the toolbar at the top here, for example, when this view is initially docked, on the right hand side, you can see it's pretty narrow. If you don't see all of the options on the toolbar, there is this black arrow that is available on all the toolbars that expands the rest of the remaining options. And so there are a couple of settings that you can set for this view. You can tell it which data to show, and you can actually make it show multiple trend lines here. So you can show the final deuteration in Daltons or in percent terms. So now I just swapped from normalized deuteration to just raw deuteration. This is the difference between centroids. You can show either the MS1 or MS2 deuteration or both on the same plot in different colors. And all of the colors are explained at the bottom of the screen here in this color section. And it has a color for the target and a color for the control if you do use a control. As well, the other options allow you to modify the size of these bubbles. So we call them bubbles because they may vary in size if you have EX1 type of exchange profile, because if you have a slow and a fast population for a peptide, they will be different sizes of bubbles. For now, everything is EX2, so they'll be the same point size across. As well as the line thickness is configurable. You can make it a little more visible by making it thicker. As well as the time scale. The time scale by default is in logarithmic time, but you can change it to linear time if you wanted to see it linearly, or even the natural log as well. There's not much of a difference between those two. The time units can be minutes or seconds, whatever you prefer your axis to show up as. And of course, you can toggle your error bars on or off. The error bars are going to be the standard deviation multiplied by this value here, the standard deviation multiplier. As well as the title is slightly customizable to include the range of the peptide or not in the title. You can show the sequence of the peptide in the title or not. And of course, as mentioned before, you can customize all of the colors on this plot. Finally, you can right-click and save this image and change its dimensions as you see fit. For example, changing it to a 1000 by 1250 height versus width.
That wraps up our look at the kinetics view and how it helps you understand protein behavior by analyzing exchange rates over time. Next, in Chapter 4, we'll focus on exporting your HDX Pro data so you can share your results and perform further analysis outside the software. Let's continue.